The overall goal of the following experiment is to demonstrate complex fluid shaping as observed by the production of microfibers. This is achieved by assembling a microfluidic channel with specific grooves to produce a polymerizable core with a predetermined cross-sectional shape. As a second step, a prepolymer solution is prepared which will be shaped and polymerized once it passes through the channel. Next, UV light is turned on at the bottom of the channel in order to polymerize the polymer material. These scanning electron micrographs show several microfiber shapes that were fabricated similarly.
Advantages of this technique in comparison with other existing methods like electrospinning and melt spinning include the fact that this technique can be performed at room temperature, and this technique can be used with a variety of polymerizable materials. In addition, this technique does not require high voltages, it does not require high temperature followed by rapid cooling times, and it also does not require control of environmental factors like humidity. Visual demonstration of this technique is critical because microfluidic fiber fabrication is not as common as some other methods, and therefore it may be difficult to conceptualize without having seen it visually.
Though this method has been used for the production of polymer microfibers, the same principles are in use in our lab for the development of other systems, like micro blood vessels.
We initially had this idea while observing micromixers and microchip flow cytometry systems, confocally, as we saw sustained ornate cross-sections develop. We posited that by introducing a polymerizable solution, the shapes could be locked in via photopolymerization continuously and with relative ease. Demonstrating the procedure will be Michael, Daniele, and Darryl Boyd.